system to work. So we are going to differentiate between general and special senses. We'll study the anatomy and physiology of the eye. We'll also study the anatomy and physiology of the ears. So that's balance, equilibrium, and hearing. And then we'll study our sense of taste and smell. So information enters the brain or the afferent pathway through all the sensory receptors that are found in our body. So we studied all these receptors in ANP1. They are called general sensory receptors and they are found throughout the body. They do not require any kind of an organ. So they only have modified neurons that's going to accept a signal. For example, as you saw that our skin was studded with nociceptors, which are pain receptors. And then we have thermoreceptors that are going to pick up changes in temperature. It's found all over our body in the skin. We have chemical receptors. They're called chemoreceptors that will respond to a chemical stimuli. And we have mechanoreceptors that respond to any changes in the cell membrane. So under mechanoreceptors, we saw the receptors that's found in the joints. So they are called nociceptors. Proprioceptors are found in the joints and they help you to make sense of where your body is in space and time. Baroreceptors will detect pressure, and tactile receptors. Remember the Pecilian corpuscle, the Ruffini corpuscle. So they are all modified neurons. So that's how we perceived. So all of them are called general senses. Our focus today is special senses. So for smell, we have chemical receptors that's found in the oral cavity. For taste, it's also a chemical receptor. We call it taste bud that's found in your tongue. For sight, we have light receptors or photoreceptors that's found in your eyes, in the retina of your eye. And for balance and equilibrium and hearing, they are mechanoreceptors. They are found in your ears. So this is what we'll be studying in the next hour. The 5 special senses are olfaction, which is the sense of smell, gustation, sense of taste, vision, balance and equilibrium and hearing in your ears. All right, so let's study olfaction, which is the sense of smell. So here we have highly modified neurons that's going to pick up the sense particle, the smell particle from the air, and then it will take it down the olfactory tract to the brain, to the olfactory cortex, so that we can make sense of the smell. So we can differentiate thousands of smells. So here's the anatomy of the olfactory system. So you can see the nasal cavity. So you can, yes?
Professor, you're still on the very first slide. We can't see anything past that very first slide.
Thank you for telling me. Let's see. Maybe I'll stop share and share again.
Is this first? I think it's your first slide. Okay, now it's okay.
Yes. Is it okay now?
Yes, it's touching, yes.
Do you see the special senses slide now?
Yes.
Yes,
all right.
We're seeing it before we put in.
Yeah.
Yeah, start me and let me know.
Okay.
All right, I think full screen is when we are having trouble. I'll just show you all like this. So here you can see the nasal cavity. And then here you can see the nostrils. It's called external nares. And when we breathe in, so the air, when we breathe in the air, the smell particles in the air are sucked in. And you can see the meatus of the nostrils. So you can see the superior, middle, and inferior concha. And these canals are called meatuses. You can see the ethmoid bone, and you can see the nasal septum here. So it's here that we perceive smell. So we have something called olfactory epithelium. So it is sticky and moist because there's many glands that secrete the mucus. And this is where we find the highly modified olfactory dendrites. See, they are hanging in there. So these smell particles will trigger a stimulus here, and the action potential will go to the olfactory bulb, olfactory tract, and up to the olfactory cortex where the information is processed. Anosmia is the loss of the ability to smell. You know, like some people can smell so well, like especially your mom, you know, they are like super smellers. They can differentiate thousands of smells. So again, that comes down to when somebody is a super smeller or a super taster, they have more of that neuron. So here, a close-up of the olfactory organ here. So here you can see the olfactory epithelium. We learned in AP1 that the epithelium will always sit on a basement membrane. So the basement layer is called lamina propria. So here we see its loose areolar connective tissue. This is where the blood supply is. The nerves are here, and the mucus secreting olfactory gland is here. So that's why our nostrils are always moist. These olfactory glands will secrete a mucus. In the olfactory epithelium, which is shown in pink here, we are going to see the modified olfactory neurons. They have all these dendrites, which is studded with receptors. These receptors are called odorant binding proteins. So see these dots here in the mucus, they will find the receptor in the dendrite. That will trigger action potential, and the message will shoot up the brain through the olfactory nerve. So what happens is they are in bundles of 20 or more. So here all these neurons are found in bundles, and they will enter through the cribriform plate of the ethmoid bone to form the olfactory bulb and the olfactory tract. So here, this picture is showing how the action potential is conveyed. So the stimulus, which is the smell particle, is picked up by receptors that are found on the dendrites. Then they trigger an action potential that goes to the central nervous system for processing. So let's see how the signal transduction pathway works for the odorant particle. So when the odorant particle finds the receptor protein in the plasma membrane, it's going to activate a G protein. This G protein in turn will activate adenylyl cyclase. That in turn will open up the sodium channels. And when sodium floods in, it will depolarize the cell. Gustation. So gustation is another word for sense of taste. Agousia is a person who can't taste well. And then like as we grow old, we kind of lose our taste buds, and we have a numbed, perception of taste. So gustation will provide information about the taste of the food that you have in your mouth, and also liquids. So here is a map of your tongue. All the sweet taste receptors are found in the tip. Salty here, sour on the sides of your tongue, and bitter taste is perceived in the back of your tongue. In addition, a recently discovered receptor is the umami receptor. So they pick up amino acids that's found in proteins like chicken broth or cheese. They pick up glutamate and aspartate. So when they say that some people are super tasters, they have a higher concentration of these receptors in their tongue. So here, what are the different types? It's also called linguinal papilla because it's a projection on the tongue. You know, if you stick out your tongue and look at it closely, you'll see these tiny taste receptors. They are called linguinal papilla. It's an epithelial projection on the superior surface of the tongue. So let's look at filiform papilla. So you can see how the papilla is sticking out. So this is not a taste bud, but it provides friction and keeps food in your mouth. It stops the food from sliding down your throat quickly. So it's holding back on food so that you can enjoy the piece of food that's in your mouth. Fungiform papilla has only a few taste buds. Valid papilla is the most important one. It has more than 100 taste buds here. The folate papilla is a type of taste bud that kind of degenerates over time. So if you take one of the taste buds, you can see that it's made of several types of cells. So you have a gustatory cell, and then you have the modified epithelium, dendrites, that's going to pick up the taste molecules, the food molecules. So they are called taste hairs or microvilli. And you have a taste pore. They alternate with transitional cells, and also there are stem cells. So the taste buds are constantly regenerating. I forgot to mention something here in the olfaction epithelia. So here, sorry, I'm going back here. So this is the olfactory epithelium, and you can see the olfactory cell, and you see the supporting cell in pink here. And then you have stem cells. So what the point I'm making here is both the sense of taste and smell have stem cells, and they regenerate every 10 days. Because like we put harsh things in our mouth, like, Dorito chips and so forth, that can harm the taste buds. So they regenerate. So what will happen is when we eat food, the dissolved chemicals will trigger the receptor in the taste hair, and then that will trigger an action potential that will go to the brain. For salt and sour receptors are chemically gated channels, whereas for sweet, bitter, and the umami, they are G protein mediated. So if you don't remember signal transduction pathway, go back to chapter 3 in AP1. They explain how the action potential is working. So here, I thought this is a fun picture here. The tongue of a super taster is like covered with taste buds. That's why they are so sensitive. They can easily pick up all the ingredients. They would taste something and they would say, oh, there's vinegar in it, there's ketchup in it. So they are super tasters. So if you're watching Chopped or MasterChef on TV, I think they are all super tasters. Whereas a non-taster, they are not very sensitive to the taste, so they kind of tend to like bland foods. So this is the signal transduction pathway. So we have the receptor cell in the taste bud, and the food is the stimulus. It's going to synapse at the dendrite, and it's going to convey the action potential to the central nervous system. As I told you earlier, salt and sour receptors, they work by channels. So here's the plasma membrane. So just when you say something is salty, it's full of sodium ions. When something is very sour, like vinegar or lemon juice, it's full of hydrogen ions. So they will pass through these receptor cells. So here's a channel protein here, and they will trigger the depolarization. Whereas sweet, bitter, and umami are going to use the G protein. So The sweet or the bitter taste is the ligand that will sit on a G protein, activating it, and that will activate second messengers inside the cell. And that is how the message is conveyed. All right, this would be the more important part of our lecture, eyes and your ears. So I want you all to repeatedly look at all these structures in the eye. Some of the names you would probably know, some are new to us. So we are going to study the anatomy of the eye. So when you look at the eye, the upper eyelid and we have a lower eyelid. So the anatomical term for the eyelid is palpebra. And you can see the eyelashes on the palpebra. And the opening of the eye is called palpebral fissure. So the V in the corners of the eye are called as the canthus. So we have the medial canthus and the lateral canthus. The fleshy part of the inner eye, medially, it's called caruncle here. The white of the eye is called sclera, and the clear part of the eye, where you would put your contacts, is called as the cornea. And you can see the pupil through which the light is going to enter the eye. And surrounding the pupil is the colored part of the eye. Blue-eyed people, it will look blue, green, black, brown, and so forth. So that is the iris of the eye. The explanation of everything I said is written here. You can read it later. So the eye, see, you can see that there's not much blood supply, especially in the cornea here. You can't see the blood supply. So it will get all the nutrients from the tears that will wash the eye. So the tears are circulating in the eye. So the gland that makes the tears is called as the lacrimal gland. So you can see how it's on the lateral part of the eye socket here. It will drain the tears through the lacrimal ducts, and then it will wash over the eye, nourishing the eye. So giving the eye nutrients, oxygen. It also has antibiotics in it, making the eye nice and clean and sterile. And then it drains into the, there is a hole here called lacrimal punctum. Canaliculi are the small canals. And then it drains into the lacrimal sac, side of your nose. It goes down a canal on the side of the nose called nasolacrimal duct. And then that's why when you cry sometimes, your nose waters, the tears come out through the nose. So these tear ducts and tear glands are very important for nourishing the eye. See, the eye is a very, very interesting organ. And like most of the information enters the body, enters the mind through our eyes. We are always observing things. So you can see like how the eye is protected in the socket, eye socket. So you can see the eye socket here. And then it has 6 main muscles. So this eye socket is made up of the sphenoid bone, the frontal bone, zygomatic, ethmoid, lacrimal maxilla, and the palatine bone. So these are the bones that make the eye socket. And there are 6 main muscles. So you can see inferior rectus. Rectus means the fasciculi will go north-south. Superior rectus, medial rectus, lateral rectus, and there are inferior and superior oblique muscles. And because the eye is under the control of 6 muscles, you can just like roll your eye, turn it left, inward, back, up, and you can just roll the eye in the eye socket. So let's look at just one muscle. So the inferior rectus muscle originates in the sphenoid bone around the optic canal. It inserts in the medial surface of the eyeball, and the main action is when you want to look down. And it's innervated by oculomotor nerve 3. And you can also, because eyes are so precious in the body, look at all the other nerves that serve the eye. abducens, oculomotor nerve 3, and trochlear nerve. They all will innervate the eye. So here in the side view or the sagittal view of the eye, you can see that the eye is not perfectly round, right? So you can see a bulge in the front. So this is called as the anterior cavity of the eye. And behind it, behind the lens would be the posterior cavity of the eye. So the anterior cavity is very small and the posterior cavity is large. The anterior cavity is filled with a watery liquid. It's called aqueous humor. So this aqueous humor is made by a structure called ciliary body. It's right there, ciliary body. And then it circulates within the eye. So the liquid, so it's one of the body fluids. So this anterior cavity has aqueous humor, which is very, very watery. It is made by the ciliary body. And there'll be a hole here called canal of Schem. So it will circulate, thereby nourishing the eye and maintaining the pressure of the anterior eye. So it's right under the cornea. See, in many diseases, diabetes related, when they say glaucoma, so it's the pressure here that they're talking about, which can be very detrimental. The posterior chamber is nice and large, and it is filled with a clear gel or jelly-like mass. It's called vitreous humor. It gives shape to the eyeball, and it supports the retina. So if you take the eye, it is supported by and protected by three important layers of the eye. The outer fibrous layer or tunic, there's a middle layer and an internal layer. So we look at the three layers of the eye. So what this picture is actually is showing is anterior cavity and posterior cavity. So let's look at the three layers of the eye. So the fibrous tunic is the outermost. So it's shown in pale blue. Some of the structure it supports, so it's tough fibrous connective tissue. So some of the structures that are found here is the white of the eye, which is called sclera. The clear part of the eye is called cornea. The junction between sclera and cornea is called limbus. And the vascular tunic is the middle layer, and that has the iris, the colored part of the eye. The constrictor muscles, that is anchoring the iris, and you can see the pupil. And the third layer would be where you find the retina. So more information of the vascular tunic. So in the vascular tunic, you will find all your blood supply. So you can see all the blood supply, and it will also have all the lymphatics. So it regulates the amount of light entering the eye through the pupil by the constriction of muscles here that will decide the pupil size, by the iris diaphragm. It's like a diaphragm, by either constricting or relaxing. You can see the circulation of the aqueous humor happening within the eye. It controls the shape of the eye. So here you can see some of the other things you can see here in this picture is when light enters the eye, it is going to enter through the pupil and it's going to hit the retina of the eye. So the shortest distance and the straight line of the path of light is called as the visual axis of the eye. So see, there's lots of things happening here. You have to repeatedly look at it and memorize all these terms. So if you study the path of the light, it's going to hit the cornea. It is going to enter through the pupil, and then it will hit the lens, and then the image is going to land on the retina. So here, pupillary muscles are the muscles that surround the pupil. So they control the size of the pupil. So you can see when there is less light, the pupillary, so there are two concentric muscles here, one muscle and a two muscle. So they will dilate, allowing more light to enter in a dim situation if there's not enough light. Whereas if it's too bright, they will contract and it will reduce the pupil size. If you were in the lab, you would have done a dissection of the cow eye, and this is how it will look like. It's amazing. It's so beautiful. You would have, there's something called tapetum. That's why cows and animals, we don't have it. Humans don't have it. So they are able to bounce light back and forth in the eye socket, and it's so beautiful, like a jewel. So you would have seen tapetum in the cow's eye. And then you can see the lens here. So the lens is made up of a protein called crystalline, which is a transparent protein. And then it also provides the focusing power of the lens and the clarity of the lens. So when this gets cloudy, is what we called as cataract, connected with aging. All right, the most important layer is the neural layer. So it has an outer pigmented part and an inner part. So we can see an outer pigmented part. So this is a microscope slide picture. And it's in the inner part where we see the famous rods and cones. So, this is cone shaped. See, this neuron is cone shaped. That's why it's called as a cone. The rods are rod shaped. So these are the cells will have the photoreceptors. They have a special protein that will receive, and it's able to receive the light. That's why it's called photoreceptor, rods and cones. Rods are very good at just discriminating light, whereas cones are very good at providing color vision. They are concentrated in one spot of your eye. That region is called as the macula. And in the macula, there is a concentrated spot called fovea. So here we see the rods and cones, and then they will synapse with the bipolar cells here. See the bipolar cells. That will synapse with the ganglionic cells. And then all of them as a bundle, they'll form bundles and they will become the optic nerve. There's other cells called horizontal cells that are found on the outer portion of the retina. And there's also another special cell called amacrine cell that will play a role. It's found between the bipolar and the ganglionic cells. So here you can see the pigmented epithelium, and here are the rods and cones. So if you zoom into the rods and cones, they have all these discs. And in these discs, if you look at the cell membrane, so this is the phospholipid cell membrane, and this is where we see rhodopsin, which is the photoreceptor. So this is the special molecule. That in turn is made up of two molecules called opsin and retinol. But when light hits the rhodopsin, there's a conformational change that will trigger an action potential in the bipolar cell, and that will convey the signal to the ganglionic cell. So all of this is very complicated, but it's also like very, very interesting. When they say vitamin A is very good for your eyes, it's because retinol, which is part of rhodopsin, is synthesized from vitamin A. And if there's insufficient vitamin A in the body, it can lead to blindness. And this is very common in developing countries. Children going blind just because of lack of vitamin A and lack of education. All right. Accommodation. See, many of you might be wearing glasses now. See, even I am wearing glasses for farsightedness because I can't. This is age related. So think of your mom and grandma who wear glasses because they can't read their books because they are farsighted. So you can buy the glasses in a dollar store because it's a biconvex lens. It's a cheap lens, and then you can fix that. So what is accommodation? So depending on the distance of the object from the eye lens will change shape. So the lens is under the control of suspensory ligaments here. So by contracting and relaxing, it can have the lens as like circular like this or flattened like this. So when you are looking at objects that are closer, the lens is circular. Whereas when you're driving, you're looking at far away objects, the ciliary muscle will relax and the lens is flattened. So thereby focusing the image on the retina. So people with myopia, or nearsightedness, that means they can't see objects that are far away. So they would wear corrective lenses because the image is projected in front of the retina. So they have to wear a lens that will correct that situation and then project it on the retina. Hyperopia is the opposite. The object is projected behind the retina. So you'll put a biconvex lens to project it on the retina. Astigmatism is another eye disorder where the image is distorted. So this is more serious. There are many videos I wish I can show you, but I want you all to watch this later. Come back and watch these videos. So let's see how the message from the eyes and the photoreceptors, how do they travel up to the brain? We saw this slightly in ANP1, the optic chiasma when you do the brain dissection. So everything from the left eye is processed on the right side of the brain, and the right eye information is processed on the left side of the brain. So beginning at the photoreceptors, the information will end in the visual cortex of the cerebral hemispheres. And what happens is, as the message goes, they are crossing over. This is called the optic chiasma. So the information crosses over. So the photoreceptors here are here. This is where the rods and cones are. They will convey the action potential to the bipolar cells. And then another synapse will happen, and that moves to the ganglion cells. And then all of them run together to form the optic nerve. And then the optic nerve will make its way to the brain. So the combined field of vision will arrive at the visual cortex of the opposite occipital lobe. The left half will arrive at the right, and the right half information will arrive at the left. So that's about the eye. So we briefly went over the anatomy of the eye and the physiology of the eye. Kindly read the lecture, study the textbook, and then just look up several videos on YouTube. Khan Academy has some, the Crash Course, they have very good videos. So I hope you'll watch all of those. All right, here. Moving on to the next special sense, which is hearing and equilibrium. So we see the ear, and this part, so it has three parts. External ear, you can see it. Then it's the middle ear, and then we have the inner ear. So we'll study the different anatomical features of the three parts of the ear. And then see this snail-like structure is the cochlea. So this is where sense of hearing is found, and balance and equilibrium are found here in the semicircular canals. We'll study that. All right, when we say external ear, you can see the pinna of the ear, right? This is where you would have your piercings, and it's also called auricle of the ear. So it has a very special shape. It's like a funnel. So it's going to grab on to sound waves, and then it will direct it inside the ear. The ear canal is called external acoustic meatus. And here we see the ceruminous gland that makes the ear wax. So the function of the ear wax is to slow down the growth of microbes here in the ear. And then we have the very delicate eardrum. You can see how it has like a concave shape here. The eardrum is also called as the tympanic membrane. It is thin, semi-transparent, cartilaginous sheet that is going to separate the outer ear from the inner ear. So outer ear, not much happening. We have the auricle, external acoustic meatus, which is covered with hair, and ceruminous glands, which is protecting the ear. It can deter microbes from growing, and the hair will keep out all the debris. People always say don't use the earbud, because if you go a little deeper, you might even rupture the tympanic membrane or the eardrum. So the middle ear starts with the eardrum or the tympanic membrane, and then there are three small bones, the three smallest bones of your body. They are called auditory ossicles. So here's a zoom in of the ossicles. We have the malleus, incus, and stapes. The malleus looks like a hammer. Incus looks like anvil, and stapes is the stirrup part. And you can see the smallest muscles of our body, tensor tympani muscle attached to the malleus and stapedius muscle attached to the stirrup here. So these muscles play a huge role in toning down. Like when you're listening to loud music, we don't want to convey all the sound waves inside into the middle ear, because that is very, very delicate. So these muscles will tone down the loud voices. Again, as you age, it's these muscles that will lose their, lose its contractibility, and that's why it can lead to, hearing loss. Also, we lose a lot of hair, which is also connected with aging. So what happens is sound waves will, sound travels in waves. So it's a vibration, it's a mechanical energy. So when that hits the eardrum, it will convey the message to the malleus. They will all start vibrating. Malleus will convey the information or the vibration to incus, and then that will convey it to stapes, and that is going to hit on a spot called oval window. So the vibrations are going to enter the inner ear through the oval window, and it comes out through the round window. This is where the action happens. This is the challenging part for us to understand. So if you see the inner ear, it is a bony labyrinth. So the bone is snail-shaped. It's called cochlea. And here it is half tubes, right? So it's called semicircular tubes. So it has a bony labyrinth, and inside the bone, there is this blue tube. See that it's not a blue tube, but in the picture it shows as blue. It's a membranous labyrinth. So there is a fluid that is flowing through these membranous labyrinth. It's called endolymph. So outside the endolymph, it will have perilymph. So if you should take a cross section of the bony labyrinth, this perilymph and this endolymph. So the sense of hearing is in cochlea. The semicircular canals, there are three of them in the XYZ axis. So they're called anterior, lateral, and posterior. So the semicircular canals have receptors that are stimulated by the rotation of the head, and they play a role in dynamic balance. Cochlea is for hearing. Sense of gravity is perceived in the vestibule. See, the swollen region here is called as the vestibule. Sense of rotation is perceived in the semicircular canals. And balance and equilibrium is in the vestibule. So one more time, cochlea for hearing. sense of rotation in the semicircular canals, balance and equilibrium in the vestibule. So what's happening is, for equilibrium, the receptors are found in the vestibule. In the vestibule, there are these hair cells that will provide information about the direction and strength of the movement of your head. So the hair cells are embedded in a gelatinous matrix called cupula. And the whole thing is enclosed in an ampulla. So, and then see, all these hair cells in turn are connected to the sensory nerve here. So the long hair cell will be called as a stereocilia. And then there's a shorter hair cells too. So the main idea here is when you tilt your head or you rotate your head, or you are going in a car for linear acceleration, or you're going in an elevator, what will happen is these hair cells will change, undergo, are distorted, they change. And that will be perceived by the sensory neuron as an action potential. The largest hair here is called kinocilium. So here you can see the hair cell is found in the cupula, right? That in turn is found in the ampulla. So when the hair cell changes its the direction because of the movement of the head, that will trigger the action potential. So they are found, so in the base of the semicircular canals, these hair cells are found. So when the head is rotating in the one plane, it will cause distortion of the hair cells. So depending on how the hair cell is moving, when you are saying yes or no, or when you're tilting your head, it will trigger the action potential. So here, there's another interesting thing that is happening here. So the swollen part of the vestibule, so you can see a structure called utricle, and there's another structure called saccule. So here there is these clusters of hair cells. So these clusters of hair cells, they are protected in this gelatinous mat. And then we have this very special feature happening here. It's called otolith. It's calcium crystals, calcium carbonate crystals. It is called, all of them together are called statoconia. So these are nothing but, it's also called rocks, you know, otoliths are the ear rocks. So here you can see when this person is upright, the otoliths are sitting right on top of the hair because of gravity. And when this person is tilting their head, the rocks will move down, move back, again because of gravity. And when it moves, it's going to drag the hair with it. That is going to trigger the action potential. So like if you think of times when you were like in a merry-go-round, you know, or like you were spinning around when you were a child, even after you stop spinning, it feels like the earth is still moving, like things are still moving, right? It's because the endolymph is moving and the hair was distorted. That's why you feel disoriented for a few seconds. And like a very good example I can think of here is, see, if you have been on a roller coaster, like in, Six Flags or Disney World, you know, one of those harsh roller coasters, you know, sometimes when the roller coaster just falls down, see, there wasn't enough time for the otoliths to come back to its normal position. They are still dislocated. That's why you feel like you're falling after the roller coaster and your head is spinning after the ride is over. So vertigo and many of the like motion sickness and stuff, they all connected with this problem here, the otoliths not going back to its original position. That's why physiotherapy helps for that. That is balance and equilibrium. Now moving on into our sense of hearing. So here in the middle ear, we have the auditory ossicles will convey the the sound waves in the round window. So if you unwrap, this is a cross section of the cochlea, the snail-like structure, which is responsible for hearing. So there are 3 membranous tubes inside. So from the oval window, the sound vibrations will kind of like go up to the tip of the snail, and then it will come out through the round window. So the frequency of the sound will determine which part of the cochlear duct is stimulated, and intensity directly relates to how many hairs. So it is here you can find hairs, how many hair is stimulated. So if you take one membranous structure here, we have, it's called scala tympani. Then this part, the blue part, is called as the organ of Corti, and then scala vestibuli. So scala vestibuli, this part is also called cochlear duct or the spiral organ, and then scala tympani. So there are 3 parts. So they are spiraling up and they come out. Another picture can show this. So in the organ of Corti, named after the scientist who discovered the organ for hearing, we have scala vestibuli, the cochlear duct, scala tympani. So this is the information from the oval window is coming in. So what happens is, in the organ of Corti, there's something called tectorial membrane, and right underneath are the hair cells. So when the sound waves leave the cochlea and then it finds its way out to the round window, it will trigger or it will displace these hair cells. So these hair cells will move it will brush against and bump against the tectorial membrane. So what happens is these sound waves are converted into an action potential, and the message will go out through the cochlear nerve. See, all of this is very challenging. I don't expect you to understand it the first time. You have to keep reading it several times until you get it. So one more time, organ of Corti has receptor cells. If you zoom into the organ of Corti, it will have a basilar membrane that houses the hair cells, and it brushes against the tectorial membrane. So when the sound waves are passing over, right, it will distort the basilar membrane. That in turn will distort the hair cell. It will brush against the tectorial membrane. That will trigger an action potential, and then the message leaves through the cochlear branch of the nerve. I want you all to watch this video here later. It's a 6-minute video which will explain how the whole thing happens. So overall, how do we hear? So here's the auricle and the pinna. They're going to direct the sound waves, and it will come and hit the eardrum here. They are arriving. So when the sound waves come here, they are translated into vibrations. I forgot to mention, in the middle ear, we have the eustachian tube, or the auditory tube that goes to the back of your throat. This plays a huge role in equalizing the pressure in the inner ear and the outer ear. So the sound waves are transferred by the 3 auditory ossicles, and then it hits at the oval window of the inner ear. So the sound waves are traveling up the cochlea, and then it comes back out through the round window. So the pressure waves will distort the basilar membrane on the way out to the round window. That will distort the hairs that are found here. That will trigger the action potential. So information about the intensity of the simulation will go to the cochlear branch. So what happens is the nerve from the semicircular canals and the nerve from the cochlear branch will come together to form the nerve, the oculomotor nerve here. So here we see the cochlear ganglion. So the information, the sensory axons from the cochlear ganglion is going to shoot up to the brainstem here. Number 2, the axons from the neurons in the cochlear nucleus will project to the alveolar nucleus or the inferior colliculi. Then it goes to the auditory cortex. So that would be the pathway of the message to the auditory cortex.